So our next topic in the biotechnology unit is gene therapy. Okay, gene therapy. So class, you might have heard about lots of therapy like physiotherapy, okay, and chemotherapy. Chemotherapy is a therapy or a treatment of a patient using chemicals, okay, and physiotherapy is treatment of a patient suffering from some kind of body ache using all kind of physical movement, okay, physiotherapy. So similarly here there is gene therapy, that means treatment of genetic disorders, okay, treatment of patient undergoing genetic disorders using gene from any other source, okay, using gene which is functional, okay, from any other source. So this all is included in gene therapy. Now it's a technique, okay, which allow the correction of gene defect by introducing normal copy of gene into the individual at the embryonic stage. So this gene therapy is performed at embryonic stage. If there is genetic disorder diagnosed before the baby is born, okay, then if the gene therapy is treated at embryonic stage, then it will last long, okay, it will be permanent. Okay, now when it is done, okay, this gene, the normal gene, take over the function of defective gene and it compensates the loss of the function of non-functional gene. So if some of the gene, okay, in the embryonic stage is non-functional, then it is treated with the gene from any other source, okay, then the gene which is normal and which is extracted from the other source is included in the embryonic stage, then it will compensate the loss of that non-functional gene and it will be permanent, okay. So genetic defects like sickle cell anemia, okay, SCID, metabolic disorders like ADA, that is adenosine diaminase, okay, short form it is ADA, so could be cured through this gene therapy. This kind of genetic disorders can be cured through gene therapy. You can look into your notes. So first gene therapy, okay, first gene therapy was practiced to 4 years old girl, okay, not in the embryonic stage, but it was performed to 4-year-old girl, and she was suffering from ADA, that means adenosine diaminase defect, okay, she was suffering from ADA and this gene therapy was done. She was 4 years old when it was done, okay. Now what did they do? That is, or we can say, how can the genetic disorder be corrected by gene therapy, okay. The process, the steps involved here is lymphocytes from bone marrow of SCID sufferer, okay. Now steps involved, these are just the general knowledge. Now how through this gene therapy genetic disorders can be cured, okay, we have to look into the steps how it is done. Number 1 is lymphocyte, that means white blood cells or lymphocytes, that is cells of cells, okay, and lymphocytes from bone marrow of SCID sufferer, or we can say any patient, okay, which they are who is undergoing genetic disorders. So the lymphocytes from the bone marrow, of a patient was grown in a culture medium. Now you know that this lymphocyte, okay, it has got one defective gene. Suppose the patient is undergoing SCID, these disorders, okay. Now this lymphocyte does not have the antibodies which can fight for this SCID, okay, these disorders. So these lymphocytes from bone marrow of a patient suffering from SCID was grown in a cultural medium, okay, synthetic medium. And the second step is cDNA of functional ADA gene is produced. Now, of functional ADA was produced, like cDNA, that means DNA, okay, DNA of functional ADA. That means if the patient is suffering from genetic disorders, then this DNA must be extracted from some other source, other organism, okay, which can have the functional ADA, which is not defective, which can have functional ADA. So from any other source, the functional ADA was functional cDNA, sorry, cDNA of functional ADA was produced, okay, the DNA which can code for this functional ADA, okay, adenosine diaminase, that was produced biotechnologically, okay. How? Through reverse transcription, because cDNA is the DNA which is produced from RNA, okay. cDNA is DNA which is produced from RNA, and if DNA is produced from RNA, it is called reverse transcription process. And if mRNA is produced from DNA, it is called transcription. But if DNA is produced back from mRNA, then it is called reverse transcription. So similarly, through biotechnological process, okay, cDNA, okay, from RNA of functional ADA was produced, okay. Now, then what happens? The cDNA of functional gene is then introduced into lymphocyte. Now, this cDNA, which is functional, which has got functional gene for ADA, is then introduced into this lymphocyte, these cells, okay, because this lymphocytes was grown in this culture, so it was introduced to that lymphocytic culture, okay, lymphocyte which was grown in a culture. Okay, this functional cDNA was introduced into these cells which was grown in a culture. Now, and this is done using retroviral vector, because in any processes when one gene is introduced into the other cell, okay, or other organism, body of other organism, always remember vector, there must be one carrier, okay, vector must be used. So here retroviral vector is used, virus is used as a vector here, okay. Now, lastly, transformed lymphocytes are re-injected into the bone marrow of patient. Now, when the cDNA is introduced into lymphocyte, then each cells of this lymphocyte, each lymphocyte, okay, it not only it has got this cDNA, functional cDNA which has got functional gene for ADA, okay. Now, these cells, all the cells are transformed, all cells are transformed because this has got the functional cDNA which code for functional ADA, okay. Now, this lymphocyte which is already transformed, now which is functional for this ADA, is again re-injected to the bone marrow of patient, re-injected back to the bone marrow of patient. So, this is how gene therapy is done. Now, when it is re-injected into the bone marrow of patient, now this patient has functional ADA, okay, functional gene for ADA. So, this is how gene therapy is done. Now, one thing remember, since lymphocytes are not immortal, that means these cells are not living for long, okay, it will die, cells will die, you know that. So, it could not give permanent solution. This therapy which was performed to 4-year-old girl, it did not give permanent solution, okay, but however, if the defective gene is corrected at zygotic or embryonic stage, it can give permanent solutions, which I have already told you. Okay, if this kind of gene therapy is done at the embryonic stage before the person is born, then it can give a permanent solution for his or her lifetime. Okay, but if the person is once they are born, and after that only if genetic disorder is detected and then the gene therapy is done, it cannot give permanent solution, because every cells inside the body of living organism, it is not immortal, it will die sometime. Okay, so it cannot give permanent solution, it is just for the temporary solution. Okay, so this is all about gene therapy. Okay, so the next topic that we are going to do is ELISA. Okay, this stands for enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. Now what is ELISA? Okay, it is the molecular diagnosis technique. Okay, it is the molecular diagnosis technique, that means diagnosis of a gene at the molecular level. Molecular level means at DNA level. So it is the early diagnosis, molecular diagnosis technique for early diagnosis or infection of diseases, which is based on antigen antibody interaction. Okay, how it is detected? It is detected on the basis of antigen antibody interaction. Antigen, you know, antigen is the protein which is produced by the pathogen. Okay, pathogen, disease causing organism, and antibody is the protein that is produced by our body in order to fight against those pathogen. So based on how antigen antibody interact with each other, okay, that it is detected, the disorder is detected. Okay, okay, now the steps involved, how it is done, how this diagnosis is done. Okay, so steps involved here is, number one, prepare antibodies against a specific antigen. So now if we are suspecting some person to have a specific disorder, we are not sure whether he or she is suffering from that particular disease or not, that particular disorder, genetic disorder or not. But if we are just suspecting, then we need to prepare antibodies. Okay, we need to prepare antibody. Antibody are protein that fight against the pathogenic organism. So we need to produce antibody against a specific antigen. Now what is that specific antigen? Is the antigen which we are suspecting. If the person is suffering from this and that disease, that means the antigen will be this. Okay, so we are suspecting, so we need to prepare antibody against that antigen which we are suspecting. Now number 2, immobilized polyvinyl chloride with antibody. Okay, so there is one instrument called microtiter. Okay, it is like this. Small cone like structure will be there, like this. Okay, and it goes on. Okay, so this instrument or a tool like this microtiter, which is called microtiter. Okay, now immobilized, that means coat this microtiter, this hollow structure, cone like structure, coat this microtiter with polyvinyl chloride. Okay, coat polyvinyl chloride microtiter with antibody. The protein that we have extracted prepare, okay, we need to coat this microtiter, okay, we need to coat this microtiter with that specific antibody. Okay, now what we are going to do is add the blood serum of infected person, okay, the person who is suffering, okay, we need to add blood sample of that person to the to this microtiter. We need to pour the blood sample to this microtiter, okay, in this microtiter, which is having antibody. Now if the patient is suffering from any diseases, then the blood of that patient will have the antigen, okay, pathogen, and that pathogen will produce this antigen. The blood sample must have antigen, and this microtiter have antibody, okay. Now we need to, after pouring blood sample in this microtiter, we need to incubate it at 37 degrees Celsius to allow the formation of antigen antibody complex. So in order to form, because this is coated with antibody, and after that we have poured blood sample from the patient which will be having antigen. So now antigen and antibody will form the complex, okay, will form the complex, will interact with each other when we put this into incubation, okay. We need to incubate it at 37 degrees Celsius. Now what happens after that? Wash to remove excess of antigen, okay. After that we need to wash excess of antigen which is not bonded to this microtiter, okay. We need to blot all that thing, and now add enzyme specific to the antigen. Now after that we need to add enzyme which is specific to the antigen, because we are suspecting that person suffering from certain diseases, so we have prepared that antibody against that particular antigen, and that antigen will be having a specific enzyme which will be bounded to that antigen, which can form complex to with this antigen, okay. So now we need to add the enzyme which is specific to the antigen, and again incubate it at 37 degrees Celsius, okay. Now if the enzyme is specific to that antigen, it binds to it and forms antigen antibody, because antigen and antibody, they have already formed a complex. If it is true, if our suspicion is true, okay, then they will form the complex. Then if the enzyme which is specific to antigen are again added, then it will form antigen antibody enzyme complex, okay. So it is called, this process, it is called agglutination. This process is called agglutination. Now add the substrate for enzyme to form color complex. Now we need to add substrate, okay, for the enzyme. For this enzyme we need to add substrate. Now after adding substrate, what happens? Add the substrate for enzyme to form color complex. That means when we add substrate to the enzyme, it will form color complex, because before that we cannot clearly see whether the antigen antibody enzyme complex has been formed or not, because it is not seen with our naked eye, okay. We need to dye it. It need to form a color complex so that it will be visible, okay. And in order to see all those things, in order to have this complex colorful, we need to add substrate, okay. So add the substrate for enzyme to form color complex, because antigen antibody cannot be seen by naked eye, okay. So this is how early diagnosis, molecular diagnosis technique is done. Now if remember one thing, if this antigen antibody enzyme complex are formed, then we can say that yes, the person is suffering from this and that diseases. But if whatever we have suspected, okay, then that the antibody which we have prepared for that antigen, okay, if it will not form the complex, then we can say that the person is not suffering from so and so diseases, it is some other diseases, okay, it is not that diseases but some other diseases. So this way it is detected, okay. If this antigen antibody enzyme complex is formed, then our suspicion will be true, okay, our diagnosis will be true, okay. So so this is all about ELISA. Now diseases like TB, tuberculosis, AIDS and other bacterial diseases, all the diseases can be detected by this technique, ELISA technique, okay, which is enzyme linked immunosorbent assay. Alright, so this is all about gene therapy and ELISA.